Welcome to the new cycle of UIB-CSIC seminars. Okay, today we have Orkun Soyer, and Marco is going to present him because it's his friend.
Yes, so we start with something that is not from a talk that is not from a postdoc or a PhD, but hopefully it's going to be interesting nevertheless. So it's our pleasure to host Orkun, who is visiting from the University of Warwick for a couple of weeks with a visiting fellowship from the university. Orkun, I know Orkun. We've been sort of chatting and collaborating for many years now. So he's done his PhD in Michigan a few years ago. Then he went for a postdoc in Zurich. First postdoc, and then he moved to trained in the Microsoft Research Center in Italy. And after that, he landed at the University of Exeter, where he became a professor very early on. And in 2013, I think, it's already 11 years, he moved to the University of Warwick with a big grant from UIB-CSIC. He's been promoting a lot of activities at Warwick and in the UK, particularly interdisciplinarity between microbiology and physics and chemistry. And recently has been awarded a multi-year very big grant from the Becky and Gordon Moore Foundation in the University of California to study things that I guess are related to the things he's going to present to us today, which is essentially structures of community, microbial communities. So I just wanted to say that he's a very approachable person. So if you want to bother him this coming week, just go and knock at the door of his office if he's there. Otherwise, he's going to be in the lab. And if you don't manage to find him and you still want to talk to him, just either send me an email or just send him an email. Sure, if you type Orkun Soyer, you will find his email. So without further ado, I'll leave the floor to Orkun. Thank you.
Thanks, Marco, for the introduction and for hosting me. It's great to be here, and thanks for coming today. I realize it's a Friday and it's before lunch, so really appreciate that you're here. And I'm going to try and keep you awake and interested. I have a lot of slides. This is my title slide that's put up behind me. And I tend to speak quite fast to go through these lots of slides that I have. I don't know if I managed to go through all of them, and some things will possibly be unclear. But yeah, feel free to stop me if it's very unclear or confusing. And then if it's a little bit confusing, I'm happy to talk more about things afterwards, of course. As Marco said, I've been at the University of Warwick for a while now, and the two topics that my group works on that I think are interrelated are cellular metabolism and microbial communities, because most of the times what we find in microbial communities are metabolic interactions. And there is a question, therefore, why metabolism is distributed in some cases and why sort of there are metabolic outputs from cells. Why don't cells keep things to themselves? And sort of on the cellular level, we try to work on that question, mostly with mathematical modeling, but also recently with experiments, mainly in yeast, actually. And the questions we ask there are related about metabolic excretion. Why do cells excrete metabolites, and how does this relate to internal metabolic fluxes? I'm not going to talk about that. That's a separate talk, but I'm happy to chat about that topic. And instead, today, we're going to talk about communities. And I put up some of the questions that we ask here, but just this next introductory slide will make the main question more clear. Of course, communities have been sort of popularized in the last 10, 20 years now with the advance of sampling techniques mainly. I mean, people started to sample different environments, and everywhere we find a bunch of microbes existing in the soil, in the gut, et cetera, et cetera. But of course, the main question is, call these microbial communities, but how much of a community is this really? Is it just a bunch of microbes just happen to be there, or are there really stable communities with a certain function and so on? And I would say that these are still unanswered questions. And the way we approach this is to treat these systems as dynamical systems, right? There are different microbes. They happen to be together at one instance that we sample, but ideally we should be sampling over time and looking at how that system actually changes over time. And on top of that, you can also add influences from the host or from the environment, depending on the system you're looking at. But this is sort of the context in which we work on microbial communities. So we really think about them as dynamical systems. And if you like to sort of read more about that, we over the years have been writing sort of opinion pieces about this sort of thing. And also here are the links to two major seminar or meetings, or rather residential meetings that we organized. Both of those were at the Isaac Newton Institute in Cambridge. Okay, so the main question that I like to talk about today is within this picture. I already mentioned that these are dynamic systems, but one point that's usually sort of disregarded is that not only are these dynamic systems, they're also usually spatially organized systems. So if you look at microbes in the soil, of course, the soil has a structure, but even if you look at microbes in a marine environment, there tend to be formational structures. And we like to understand how these, what the effect of this structural formation is. And this is just to say that people have already looked at the spatial organization, but mostly in model systems. For example, biofilms are very much used to understand the effects of space on interactions. And we have done some work on this as well. But one specific work I like to highlight is from Sunil Luxman's lab in ISTS in India, where they found actually in a single yeast colony, you can actually emergence of multiple strains that are metabolically behaving differently. So as a spatial system develops, there can be actually feedbacks from that spatial formation onto the behavior of the cells within that system. So even in a single species system, you can have differentiations emerging because of space. And I really like this this paper, and this leads to this idea that there are maybe feedbacks that as microbes grow, they change the environment, and as they change the environment changes them. And of course, what will be quite interesting is to try to do this in a community context where you don't have a single species but many species. Now, of course, these are interesting things, and people have been interested in these questions. The issue is how do you study this stuff? So in the nature, we find spatial organization quite a bit, and one of the systems that people have studied is, I'm just going to use my hands, I can't figure out the laser, are cyanobacterial mats. It's fine. But there are other structures as well. So these are called pinkberry structures, which contain sulfur cycling microorganisms. And then you're also like sort of grandiose biofilms that we might see here thereabouts on sort of marine environments, usually containing cyanobacteria. These are micro scale systems, basically. So these are sort of millimeter to centimeter scale. You also have structural organization at micro scale. These are again examples of cyano actually. The top one is Trichodesmium. It's a marine species mostly. The lower one can be both marine and freshwater. And they tend to form these colonies, these structures, which can also get quite large. And again, so how do you study these types of questions? People tend to go and do sampling in the environment and try to make measurements in the environment. Many of these systems are difficult to replicate in the lab. The most work has been done on cyanobacterial mats, but that's quite old now. Somehow that field has not progressed, and sort of probably due to funding issues and things like that. So we've been interested in these types of questions about how does the spatial structure emerge? What does it do to the existing microbes? How do microenvironments emerge, and so on? Again, some of these questions have already been looked at in some contexts. So what we ended up working with is what we call the cyanobacterial aggregate, a little bit like these pictures are shown here. These are like 50 micron is the bar here, if you can't see it. So something like that, where cyanobacteria sort of aggregates, but in our case, the aggregates can become sort of millimeter sized And these we first picked up from a local reservoir just by chance, really. And then this whole story started as a Friday project. Initially, we were keeping these granules in the lab with occasional subculturing and so on and so forth. And finally, 2020, the gods sent some money my way, and I could sort of start really properly working on this. And then from that point on, we started regular subculturing of these systems in the lab. So we don't necessarily relate our lab cultures now to the ecology and the real world, because it's not quite a one-to-one connection, but some connections can be drawn. But we're just treating this as a lab model system, if you wish, that forms structures, because these systems reproducibly form structures in the lab as we grow them. That you see here these examples of sort of we call aggregates or granules. There are sort of biofilm-like structures as well, and they're quite extensive. So these cluster about 5 centimeters or so large, just to give you a sense of the scale. As we kept these systems in the lab, we weren't necessarily cryostocking and going back to the cryostock, we were just subculturing them. And of course, evolution is happening. And then a lineage emerged where we don't see these structures, and instead we see homogeneous cultures as well. And we know by now that there are mutations in the dominant cyanobacteria in this system, in this homogeneous case. And I'm going to talk more about that, but there is only a single cyanobacteria in these cultures. We tend to explain them as enrichment cultures. If you're a microbiologist, microbiologists have been doing enrichment experiments, which is pretty much what we're doing here. So keeping things in the lab under a specified condition. So where this got really exciting for me is when we were taking these kinds of pictures, we also started to sort of make multiple pictures and so on. And at some point, quite early on, actually, we realized that these things are moving. And that is quite an amazing observation, at least to me, because this is not somebody slamming the door of the lab or something. This move is over a day, so the movement is quite slow, but there is a very sort of specific directed movement of these large structures. And my sort of response to that was, what is going on here? In a cyanobacteria are sort of micron scale, these structures must have millions of cells, and how do they move, like as an entity? And at this point, the project has split into 2 parts. We wanted to understand some of those questions that I already posted, sort of more microbial ecology questions on one hand, on the other hand, we wanted to understand the physics behind this. And of course, the 2 things are interlinked. Okay, so my talk is divided into 3 parts, and then there's a prologue as well. So, and I already talked a lot, so I don't know if I'm going to go through all parts, but let's start. So the first part was about, as I said, how do these aggregates actually come about or move? And we tried to first understand the cyanobacteria, because as I said, we got a lineage that doesn't create these structures. And the mutation in that system is in the cyanobacteria, and the cyanobacteria in that system is not motile. So that suggested to us that this whole structure business is related to the motility of the cyano. So we want to understand that first. And when you put these guys under the microscope, the cyanobacteria we have is a so-called filamentous cyanobacteria. So individual cells are visible here, and at division they don't separate fully, so they form these chain-like filaments, or what people call filaments. If you look at them under the electron microscope, you don't see any flagella or pili type structures, which is quite curious because normally motility is linked with flagella or pili in bacteria. So therefore, this is a bit of a puzzle, how these bacteria move, how these cyanobacteria move. And in terms of their movement characteristics, they tend to go back and forth on sort of very well-defined trajectory. So we are looking at, this is a low magnification microscope image. We are looking at individual filaments in that movie. So people have called this a gliding motility, if you look into the literature. And as I said, the molecular basis of gliding motility is still not quite fully understood. The poster model organism for gliding motility is Myxococcus, which is a single-celled organism. And Tam Mignon's group in France have worked a lot on the system along with other groups. And their proposed model is that the cells have a cytoskeletal feature to them. And on this cytoskeletal element, motor proteins can travel around. And because the cytoskeletal element is helical and these motors are traveling on it, as they sort of reach the interface between the cell and the substrate, they will translate their own movement to the push of the cell forwards and sideways. So the cells are rotating as they move in this model, and then that's also what they're observing. And they were able to label these proteins and show that these proteins are really moving about in a helical fashion across the cell. It's still not clear how this transformation of transport of force will occur, and where these proteins are exactly in context of membrane and things like this. So it's still ongoing work. Now, of course, when you come to filamentous cyanobacteria, you have multiple cells connected to each other. So it's not clear if this model could be applicable one-to-one. It could be that there are other mechanisms working, and two mechanisms that have been proposed is one, I should have mentioned that in both cases, there is secretion of slime from these cells. And in the Myxococcus case, people think that this slime allows for better attachment between these motor proteins and the substrate. In the case of cyanobacteria, people thought initially that maybe this slime secretion itself can move the cells, but we think now that's not really happening. And then an alternative is that there are pili involved, just like in twitching motility, where pili is extracted and then anchored somewhere so that the cells can pull themselves up. But again, how this model can apply in a filamentous organism is unclear. So, these studies aside, we are studying here a non-model organism, so we don't really have genetics and so on. So we thought maybe we can start with just looking at the dynamics, because many of these studies are actually molecular biology studies. So they've tried to figure out which proteins are involved, etc., but they don't necessarily look at motility, right? So the information on motility was quite sparse. And we thought that's something we can do, also collaborating with people like Marco. And then I already showed you this movie where things are moving back and forth. What I didn't tell you is that this movie is on an agar path. So what we then realize is that if we go to actually a glass slide, the movement is relatively different, but there are some key features that remain the same. So the cells tend to still go back and forth. Their trajectories are less defined, so they tend to sort of turn about. And this turning can be quite extensive at times. And you can also see that some of these filaments are seemingly detaching from the surface and then sometimes using traction. But we sort of focused on the agar case because it gives us nice trajectories, and then we can use image analysis to figure out, for example, the speed of the cells and the movement dynamics. So as these cells move back and forth, we can track their position, we can calculate their speed, and there seems to be some characteristic features to this movement. Of course, it is oscillatory, but also if you look at these trajectories here, which show the distance traveled in gray and the speed in yellow. So they travel some distance and then they stop and the speed goes to 0 and then they reverse and they travel in the other direction. So you can actually see this gray marks on this graph, which show the time that they spend sort of stopped during these reversals. And you can see that this time is not constant, so it changes between different reversals. So we can analyze the statistics of this to some extent. And two things that we found is that if you look at different filaments across their different lengths, the speed does not seem to correlate with length, which suggests to us that the forces involved are possibly shared by all cells or most of the cells. Because if only one cell, say the head cell, was pulling the whole filament like a locomotive, we would have expected a dependency on the filament length. And then this stoppage time that I was explaining to you seems to have this characteristic, sort of a characteristic exponential distribution. And now this kind of statistic, you can start to ask questions about, well, what kind of mechanisms will generate this kind of behavior. And of course, many different physical mechanisms could generate this kind of data. But we have explored a few different models, and one of the models that we found can reproduce this kind of behavior is if we assume the cells to be connected with each other through springs, and if each of the cells is producing a force to move itself forward. So we come up with a model with Marco and Emanuele Locatelli in Padua, and also Chantal Valeriani in Madrid. And we can sort of put equations here into this model and then plug the whole thing into a computer to run basically simulations of moving filaments. And the key features of this model is that we assume things are connected and by springs, so they can have a sort of a mechanical interaction. And then we can actually parameterize the sort of the strength of that interaction, if you wish, by the spring constant. We also assume that cells create a force that has a direction. It's given by this function S here, and that cells can decide pushing that way or this way. Okay. And then with this kind of model, you can actually look at what kind of parameters will make the model behave like the experiments. And here we have some parameter scans, for example, for this spring coefficient, which determines the mechanical coupling between cells. And of course, if you change these parameters, the model behaves in different ways. So these are now sort of pictures like an experiment, but from simulation, where we again look at the distance traveled and the speed. And you can see that some of these graphs very much look like the experiment, like this one, and some of them look quite sort of noisy and so on. And you can also like watch the model do its thing. And again, some of the filaments in some parameter regimes pretty much move like the experimental observations, and sometimes the movement is quite erratic. So it boils down on how cells can coordinate their force generation as they move. And if everybody is well coordinated, which happens when the mechanical coupling is big, we all pull in one direction. And then if we hit something, as we do in these simulations, actually we use a boundary, hard boundary, to make these simulations. The front cells switch their direction, and that sort of goes as a wave across the filament, and everybody switches their direction and we start to move this way. And in these simulations, that coupling is a bit broken, so cells can sort of abruptly change their direction and sort of work against each other. So basically, the take-home message from this exercise of model analysis is that there are some parameter regimes where things seem to behave as we see experimentally, and that parameter regime is characterized by strong coupling and so on. So what the model suggests is that the cells should have a high coordination within a filament. So we then want to see experimentally if that's the case, and that required us to look at individual cells as the filament moves. So we had to do high magnification microscopy, but where you can see the cells, but you cannot really tell how the cells apart so easily. So we used something called turf microscopy, which uses an angled light source, which allows you to image only 100 nanometers or so into the substrate. So you can look at the surface of the cells, and to our luck, we just happened to find that these cells have some fluorescent protein complexes on their surface. And that's something possibly relates to photosensing that we are trying to understand further. But you can see in this image these dots, and these dots are not labeled proteins, they're just fluorescent protein complexes that are naturally there. And their presence allowed us to very clearly and nicely track individual cells. So if I play that movie, we happen to catch here a reversal actually. So the whole filament was going downwards. I'm going to let it play again. So it's coming down towards me and then stops and reverses and goes back. And you can see because of those fluorescent complexes very clearly that these filaments are actually rotating as they are moving. So this sort of aligns with the sort of the Tom-Mignot type model that I was explaining before. So now I can track individual cells and I can look at their sort of position as they move and their speed. And what we find is that they are actually super well coordinated. So throughout this movement, all the cells are behaving in the same way, as if they're one single cell. But we had sort of noticed, I mean, this analysis is sort of error prone and so on, but we noticed in some cases that as if some cells are out of sync. So here I try to illustrate this actually at the reversal point, the top cells seem to be starting to turn just slightly ahead of the head cell, but it's hard to say if this is real or if it's an error, at least in this data. But we then found, so that sort of suggested to us that maybe there is a breakage of coordination at some times, cells can be decoordinated. And actually we did find this in longer filaments. So the movie I was showing to you was on a relatively short filament, and longer filaments are a bit difficult to find and film in that way. But we did find longer filaments at times, and we couldn't sort of image them with turf, but we could image them at low magnification. And lo and behold, these filaments can actually get decoordinated. So it's not easy to see it in this movie, but if you analyze the ends of these filaments, this filament, you can actually show that at some point the two ends are going against each other. And we can sort of, again, track the cells and verify that. So this is evidence that the coordination breaks and the cells can misbehave basically and lose this coupling or communication that they have. Now, as you saw in that movie, I don't know if you noticed, so if I play this, there is this sort of formation here. This is known as a planktoning formation, and it's observed in DNA. So the filament there twists upon itself, okay? And we think that this is related to cells rotating. And actually, we observed this planktoning formation in many instances. And when it happens, it can actually sort of create this sort of dragging phenomena, and it can also drag other filaments. And you can sort of get entanglements around this plankton. So we think perhaps this sort of relates to the aggregate formation and some of the aggregate dynamics, but that's something we're trying to explore. Okay, I'm going to switch gears. How long has it been? 30 minutes? Yeah. Okay. Let's switch gears and talk a little bit about the microbiology side. I already told you these are actually, we are having a community here, and the cyanobacteria is not on its own. And we've been exploring this with Sarah in the lab. And the idea was to actually do sequencing over time. So as we subculture these cultures, we take samples and we sequence them. And we wanted to see which species are there and how do they change over time in terms of population size. Firstly, what we find is it's quite surprising. We only find about 17, 18 species in the system, and some of them are unique to the non-motile lineage that emerged in the lab. And also you can see that some sort of order or genus of bacteria or families of bacteria are missing in the system. And then the other surprising finding, at least to us, is that the composition is quite stable over time. So, and when I say over time, we've been subculturing this over 2 years now. And it seems like the system has sort of stabilized and we are not losing any more species. We assume that we started with a much more complicated community, although in this instance, we don't have the starting community. I'm going to tell you a bit more about that later on. So we have something like 100 generations, and these 18 or so species are staying together. They're not being lost off the system, which raises the question of, you know, how is that possible? Why don't we have just one bacteria dominating these cultures, right? And I should mention that as we subculture these systems, we do not put any significant carbon source, and therefore we assume that actually any carbon produced must come either from dead cells or come from the cyanobacteria, which can of course photosynthesize and fix carbon. So this is just another way of presenting which species are there. Actually, we have three main families, and actually each family is represented by a number of species. So there are 9 alpha proteobacteria, only 2 gamma proteobacteria, and 5 actinobacteria in the system. So now the question is, how do they coexist? What do they do? Are they interacting, or are they sort of adapted to specific environmental niches? And to do that, we create from the genomic sequencing metagenomes. And in this case, we use long as well as short read sequencing. We also use long read sequencing. So we have high quality genomes for all the species in the system. And once you have that, you can look at which genes are there. You can tell what kind of metabolism these guys are doing. And that's led to these findings. So interestingly, all of this group have anoxygenic photosynthesis genes, which was quite interesting to us. So anoxygenic photosynthesis is a type of photosynthesis that doesn't use water as electron donor and therefore doesn't generate oxygen. It uses other electron donors such as hydrogen sulfide and so on. And I would say it's sort of a understudied form of photosynthesis. Then several species from these two groups, they have carbohydrate degrading pathways around galactose as a monomer and similar compounds. And then we have this hydrogenophaga, which is a known sort of sulfur oxidizing bacteria. And I should note that both anoxygenic photosynthesis, it's a bit less clear there, but definitely the sulfur oxidation require anoxic environments. Okay, so those functions will not be functioning in presence of oxygen, which is a bit curious because of course photosynthesis also generates oxygen. But we first wanted to sort of analyze this degradation pathway aspect, and I mentioned already the gliding motility and slime formation. So we can actually see these slime a little bit on the microscopy. We were also able to label it and find that it is actually creating a sheet around the cyanobacteria filaments. And basically we were able to extract some of this slime and analyze it chemically. And what we found, it contains so it is a multi-monomer. It's a polymer, basically, it's a sugar polymer, but it has multiple monomers. And the main monomers seem to be galactose and glucose. And this aligns with these galactose degradation pathways. And we were also seeing some evidence that some of the bacteria can, actually this movie is playing on its own. So you can see the filament here, and then all of these guys around it are bacteria attached to that slime. And we can also see some of that in EM images. So there could be some attachment on the slime by the bacteria, and we think there could be some degradation of it as well. So we were able to isolate some of the bacteria and grow them on galactose, which also align. But these are not 100% proof that this degradation is happening, but likely source of carbon is this slime within. The other point I mentioned is the oxygen aspect, and we wanted to measure oxygen gradients in these structures. So as I said, some of the aggregation forms these granules, and then we are able to fish out those granules and use microsensors to make depth profiles on them, and we're able to measure oxygen. And what we found is that actually the insides of those aggregates, even the small ones around a couple of millimeter large, can be anoxic, completely anoxic. And then I didn't mention this, but just looking at the genomes, we saw that some of the species were lacking vitamin biosynthesis pathways, and some of them had them in full capacity, especially the Pseudomonas species can make most of the vitamins, including biotin B7, vitamin B7, which can only be made, it seems, in terms of the gene content by this species. So that suggests that there is maybe some vitamin exchanges in the system as well, and we could experimentally prove this by isolating the Pseudomonas and one of the species that doesn't have, according to the genome, these biosynthesis pathways. And lo and behold, this Rhizobium rhizo species doesn't grow if you don't provide vitamins in the media, but if you provide B7 or the supernatant from the Pseudomonas, it can grow. So this sort of leads us to sort of a working model about sort of the interactions in the system in terms of metabolic exchanges, but also anoxic, oxic environments, which can maybe create some microenvironments for the different bacteria to survive and be selected for. Okay, so I'm at 39 minutes. I'm aiming for 50 minutes. Is that okay? Yeah. So the third slide is, so I'm summarizing things as I go along. So I talked about these aggregates and how it could affect the interactions, but how do the aggregates form? Right? We were quite curious to understand how the aggregation process actually happens, and whether sort of the emergence of aggregation has some impact as well. And one explanation you find in the literature is that there are sort of hydrodynamics in the environment that will lead this to aggregation. Maybe cells are being pushed together or they attach and so on. And if you actually shake the cells at different intensities, you do get different types of structures. So that definitely there's an impact of sort of environmental forces on this aggregation. But we have discovered that there's also an additional element, which is quite curious. So in order to understand how the aggregation starts, we start the film cultures from very early on. So when we make a subculture, let's say we put them in a beaker, and then we try to film these beakers to see what is going on. And in this case, the dilution is not very high, so there are already some clumps of cyanobacteria. And in this particular movie, we have some beads as well in the system that ended up floating. So this guy will sort of float above. But we are looking where these cyanobacteria are is actually the bottom of the beaker. And I'm just going to play this video. Hopefully you can see. Some of these clumps are moving, and some individual cyanobacteria start to sort of aggregate here and there. But about now, hopefully what you can see is that there's emergence of these yellow particulates in the system. And some of those yellow particulates are sort of moving about as well. So I can zoom into a part of this movie to better show you what is going on. And what we think is happening is that these yellow particulates, as they form, they are being sort of attached to the cyanobacteria, or rather being almost collected. So I don't know if you can see this well here, but all of these little bundles of green are actually cyanobacteria filaments, not individual filaments, because we wouldn't be able to see those, but possibly hundreds of filaments that are sort of somehow pulling those structures together. And we have extensive movies of this by now, and we're trying to characterize this behavior. But you might now also ask what these yellow particles are, and we found that actually, we can sort of follow this over time. What ends up happening, the movie I'm showing you here, actually, this movie is over hours. So let's just play that again. So this is over hours. Every 10 minutes or so, we are taking a picture. But we can follow this over weeks, essentially. And when you do that, you find actually this kind of collection behavior extensively continues, and then larger and larger granules form. When you then cut them open, like if you take this big granule and cut it open, the inside will be filled with this yellow particle. So it turns out these are iron minerals. So of course our media has iron, and iron can readily be precipitated chemically, abiotically, but we found that actually there is biological enhancement of iron precipitation in the system. So if we just let the media sit, it doesn't precipitate, as we see here. That suggests that either cyano or other bacteria are facilitating that precipitation process, and then subsequently it is being collected. And we can do this in different ways. We have also done SEM-EDX, which you can go with the electron microscope and look at individual particles. And we also have collected them and characterized them chemically. So iron, this type of iron cyanobacteria relation has been shown in trichodesmium as well. There is one question there about if the iron minerals can be dissolved again to be utilized as an iron source. One way that this can happen is through siderophores. So we looked at siderophores in the system, which are basically iron-chelating proteins, and our cyanobacteria did not have them, but our bacteria do have them in terms of gene functions, but also now experimentally we find evidence for siderophores, which we are trying to characterize. Okay, so basically this aggregate formation somehow interlinked with particle collection, and in this case particles are iron. And this can then introduce other functions in the system, such as iron cycling, but also electron transport among different species, or handling ROS and things like this. So basically where we are is now that we think that these filamentous cyanobacteria can possibly create a microenvironment or spatial structure in which certain bacteria can be fitted in or selectively collected into. So that's our sort of hypothesis. If this is true, this kind of phenomena should be happening more widely, and our sort of model system, what we are seeing in it, should be perhaps replicable in other contexts as well. So one question is, do we always find the same type of bacteria? Do we always find maybe iron collection and so on? So this is something now we are trying to do to more systematically repeat these experiments of subculturing. So we go out and take samples, again, from the original source, which is a local reservoir, as I said, and then we create new cultures in the lab and see if we see a similar dynamics in terms of microbial composition, spatial structure formation, and so on. And these experiments are ongoing, but yeah, I love these pictures. Some of these structures, cultures have already formed structures, and we can already start to characterize them. And actually, in one of them, we found the same species again in terms of cyanobacteria, and some others have other cyanobacteria. Some of them have multiple cyanobacteria in them, and also we do see a reduction in the community. So there is some sort of selective retainment going on. So these are the numbers of species that we see in the initial culture that we set up from the environmental sample, and these are the numbers that we have now. But these cultures have been kept only for about a year or so, whereas our other culture has been kept for 4 years or so. All right, and that's also why I am here, partly, is to try these kind of experiments, perhaps here as well, with some of the samples that we can take here. And of course, you can do this experiment with any kind of sample of your garden pond and so on. But yeah, so this is essentially why I'm here. And yeah, thanks for your attention. Any questions?
I have many questions. I will just ask you. You mentioned early on in the talk about the movement of the aggregates.
Yes.
I don't know if you have any idea of how they can coordinate such huge numbers into.
Yeah.
And if these movements are directed.
Yes. So yeah, so there is a new postdoc joining, Rebecca Poon, and she's going to focus on this basically. Our running idea is that as they get entangled, their movement, basically this is how the movement is. So some parts get entangled, they can't move anymore, but their other parts can move, and that translates into a push or pull. So these moving particles, large part granules, they are always on the surface basically. So I think what my hypothesis is that there are long filaments, and then some ends of it cannot move, but the other end keeps moving, and then this allows you to move. Again, yeah, I mean, if you have good ideas, I'm very interested. What we are trying to do now is to try to characterize the speed, for example, of those particles, whether they're directed motion. What we can see so far is there seems to be different modes. So I don't know if you notice in the early movie, yeah, maybe I don't go back now, but there are sort of granular types that tend to move around, but there are also films. So we do see that these granular parts, for example, move, and then they stop, and then they tend to form films. And then the opposite happens sometimes as well. The films sort of coalesce and form an aggregate and start to move. So we are trying to now understand, how those phases are generated. It almost seems like phase transitions of some type. But, sorry, and there was another question.
So you know if there's any kind of taxes in these?
Phototaxis, yes. So yeah, so basically there are papers saying there's phototaxis, there's photophobia. The most recent paper on this found that they align to light-dark borders. So we were able to reproduce that a little bit. And what we're trying to work on now is a single field, because most of these experiments were done with basically populations. You know, they have a population on agar and they took light and see if 2 days later they're there. They do that. So there's some phototext, but we are trying to understand how it works at a single filament level. So we're taking those filaments and film them under dark light borders and so on. The results are not conclusive so far, but we do see change in, so there is a reversal type of response basically to light. And we think that that's how they can achieve some sort of taxes. Yeah, sure.
So you showed a video in which the two end of the bacteria
Yeah.
were pulling each other apart.
Yeah.
And you also mentioned that they rotate while they move,
Yeah.
right? So what happens when they are pulling each other? Did you see the rotation in two ends of the bacteria pulling?
Yeah, so that's why we think that this planktoning is happening basically. And a student has, I have not a slide for this. But yeah, so here, basically, rather than pulling apart, what is happening is that these two ends are going against each other. So normally, all the cells, let's imagine that all the cells go forward in the context of the filament, and this whole thing will move that way. And if they're all pulling this way, this whole thing will move this way, right? But what we think is happening in this movie is that at some point, these guys are pushing this way and those guys are pushing that way. And that's why the middle buckles, but not only buckles, but it also twists, right? And we think that this twisting comes because this filament is rotating at the same time. So you have a basically elastic rod, but it's rotating at the same time as moving.
So the rotation at two end are in different directions, or is it?
The rotation is coupled to the translation. So if I'm moving this way, I'm rotating clockwise, let's say, and if I'm moving that way, I'm rotating anti-clockwise. So you're twisting the whole thing if you go the different directions. And there's something called Casarat rod model. Correct me if I'm butchering it. But this is basically about modeling an elastic rod in 3D. And people show that this can generate planktonines and so on. So we think that maybe that kind of model will be the appropriate model to figure this out. And we're thinking that maybe we can use that to find maybe there's a limit length before which this doesn't happen and that sort of thing.
Yeah, I'm just curious of how do they become entangled actually? How are they growing that way so they become filamentous, or they are just different cells that eventually become entangled?
So what they get entangled is full filaments.
Uh-huh.
I don't know if I have a good movie here, but I think if we maybe look at this one. Oops. Yeah, so here, for example, you had two filaments next to each other. It's not very easy to see, but what happens a lot of the times, multiple filaments get next to each other, and they sort of align. But again, this we see that a lot on agar, for example, where the movement seems to be confined somehow. If I play this movie, you can see that there's. Oops, so there's sometimes detachments like this guy here, like sort of almost loses contact and then swings about. So we then saw movies where that sort of thing happens and then maybe connects to other filaments. And then this planktoning, when this planktoning happens, that then again confines the movements and pulls other filaments, etc. So our sort of working idea is that those kinds of things are eventually leading to larger aggregates when you have high density. Yeah.
I have a question about the communities.
Yes.
I think that's so cool.
Yeah.
So you have seen that there is this community that is more or less stable, and then they exchange things, and that's why maybe they maintain. And you said you are going to collect more samples to see how other communities are. And I wonder whether you could create new experimental communities by mixing different kind of bacteria and then see what happens, because it would be very cool to see how diversity and functional diversity and dominance and these kind of things affect the stability and the behavior of communities in different contexts.
Yes, yeah.
So are you
Beautiful.
doing that?
Yes, that's exactly, so I'm trying to write a grant on that sort of thing. Yeah, that's exactly where we want to go. I guess you're right. You can just put things from scratch, basically, and people done that a little bit, and we have done that sort of thing in a past project. That will be interesting to see if you can complexify it enough if you started from scratch. The other idea we have is to add things or remove things from this system. For example, if I try to kill out 1 or 2 bacteria in this, will it collapse or nothing will happen? Or if I put E. coli in here, what will happen? And then the third step is now we are starting to get other sort of simpler systems. So we have one system where we found 12 species. So it somehow very quickly lost diversity. And then, you know, if you achieve that kind of low level complexity, then you can mix those cultures as well. And you can see, you know, then yeah, if suddenly you have 10 community and then you have a 20 species community, how's that going to behave and so on. Yeah, so yeah, and
it's physically possible to create them.
so yeah, no, that's the issue. So we have now in this system we have 18 species and we have 4 isolates.
Okay.
So I don't think we can go all the way, but you can try, like with those 4 species, for example, we can try to put them together. The main problem we've been having with this system is that we could not isolate the cyanobacteria. So, and I guess you want to start with the cyanobacteria. But of course you can, well, because we want to have this no carbon scenario that facilitates, I think, interactions. Although we have also run experiments now by adding glucose into the system. And depending on the concentration of the glucose, the system remains stable, it seems. Yeah, so I think it gives you, if you want, a model community to play with ideas, right? Perturbation experiments. Evolution is something we haven't looked at,
That's super cool.
but yeah. Lunchtime.
Okay.
All right, thanks a lot.
Thank you very much.